Even if I know that tomorrow the world will go to pieces, I would still plant my apple tree, hoping that one day they will be able to give the fruit. So the continuing the bacterial physiology, oxygen requirement or the aeration, on the basis of that bacteria are divided into two, aerobic or anaerobic. Aerobic bacteria require proper oxygen for balanced growth. If inadequate oxygen is there, growth is restricted. They are of following three types. Obligate aerobe, as the name obligate, one can understand they are obligate, need absolute requirement for the oxygen. In the presence of oxygen, they do oxidative phosphorylation, not fermentation, for their survival. Examples are the cholera, Vibrio, Pseudomonas, Mycobacterium. They need oxygen. Then facultative anaerobes or anaerobes. They prefer anaerobic condition, but they can also grow aerobically. They can grow in both way, but they prefer oxygen-rich condition, but also sometimes can grow anaerobically if the conditions are there. Example, staphylococcus, bacillus, intestinal rods like E. coli, they survive in oxygen, can do both fermentation and aerobic respiration according to the environment in which they are need and according to the need they are. If in oxygen presence, anaerobic respiration is stopped and bacteria respire aerobically. Microaerophilic organism, they grow best in the low oxygen tension, that is about 5%, but they are killed in very high oxygen tension. Examples are Campylobacter species, Helicobacter species, and Treponema pallidum. Then anaerobic bacteria, they grow in the absence of the oxygen and their growth is inhibited by the oxygen tension. Examples are Clostridia, which cause tetanus and gas gangrene, grow in the absence of the oxygen. They multiply in dead anaerobic tissue of the wound, produce toxins causing tissue damage. They multiply in oxygen-free environment of the vacuum sealed can of the food, produces lethal toxin of botulism. Anaerobic condition can be produced artificially by the gas bag system in the laboratories. Here hydrogen reacts with the oxygen to form water, creating oxygen-free atmosphere. And here you can see anaerobic bacteria jars, Macintosh filled jar, and gas bag which is kept there, which absorbs the oxygen. So we are able to have anaerobic environment inside these jars. Anaerobic bacteria are of two types. Obligate anaerobes, again the name is obligate. They are inhibited or killed or die on exposure to the oxygen. And these are Clostridium tetani, Clostridium perfringens, and Bacteroid fragilis. They make cell energy not by the oxidative phosphorylation but by the fermentation. As they lack certain enzymes responsible for the oxidative phosphorylation, so have to trust by mechanism of fermentation. Aerotolerant anaerobes, they can tolerate or survive in presence of the oxygen, not killed by the oxygen as obligate anaerobes, but they respire anaerobically. The third physical factor is the carbon dioxide. Our bacteria require small amount of the carbon dioxide for their growth. Bacteria get this carbon dioxide usually present in the atmosphere, or the bacteria produce it endogenously by cellular mechanism. Some bacteria like Brucella abortus need higher level of the carbon dioxide, that is about 5 to 10% for their growth. So they are called capnophilic bacteria, needing more carbon dioxide, and their examples are Neisseria and Streptococcus. Here one can see the liquid media in tubes, and that has been sealed by the corks. And then there are the petri dishes, solid media in petri dishes, and all these which need the carbon dioxide. We have put a candle inside the jar, closed jar, and that when burning is going to produce the carbon dioxide, and that is going to be used by this liquid media as well as the solid media. Moisture, water and drying. Moisture is essential for the bacteria growth. Water is essential ingredient of the bacterial protoplasm, and drying is lethal to the bacterial cells. Treponema pallidums are highly sensitive, grow only in the moist environment, causing the syphilis, while staphylococci can withstand dryness for months. So these are the tough bacteria. Bacterial spores are resistant to the desiccation and survive in dry state for many years. And freezing, drying or lyophilization, drying in vacuum and cold, is also a method in which the bacteria can survive without the water for a longer time. That is to preserve bacteria, viruses and other labile biological bacteria viable to be used in the future. pH or H ion concentration. Different bacteria need different pH for their optimum growth. Most pathogen bacteria grow best at neutral or slightly alkaline pH, that is pH of the human being, that is 7.5 to 7.6. Some bacteria can even grow in the quite acid or alkaline condition in range of the pH 5 acidic to the range of pH 8 basic. pH optimum is near neutral, and near neutral is pH 7 or the body pH. And on the basis of the pH value, bacteria are classified into three types. Number one, neutrophiles. Their pH is 6 to 8. Cytoplasm of the most bacteria has pH near 7, so majority grow optimally at neutral pH. Human blood and the tissue have pH 7.2 to 7.45, so that is favorable for these kind of bacteria to grow well in a human being. Now, alkalophiles, pH 9 to 11, bacteria like Vibrio cholera can tolerate acid condition as below as 20 and alkaline condition as high as 9.5. Proteus grow well at the high degree of alkalinity. So this is a very good way to check the pH of the urine, and if the urine is alkaline, there is the possibility of the Proteus, and if the urine is acidic, there is no chance of the Proteus to grow there. So this is a point of differentiation to see whether the bacteria is E. coli or bacteria is Proteus. Acidophile, pH 1 to 5, majority of the bacteria do not grow at this acidic pH. Only acid-tolerant bacteria grow well in acid media that is used in the food and the dairy industries, like certain lactobacillus and streptococcus species converts milk to the buttermilk and cream to the sour cream. Yogurt has acidophilic bacteria. Species pose no threat to the health. Acidic food like citrus fruit, tomatoes, vegetable cannot be attacked by the common bacteria because of this acidic condition. Then the other parameter is light. Darkness is favorable for the bacterial growth except phototrophic bacteria. Phototrophic, which like light, bacteria are sensitive to ultraviolet light and other radiation because ultraviolet rays of sunlight or a mercury lamp are bactericidal in nature. Ionizing radiation also kill the bacteria. Exposure to the light also influences the pigment production, and photochromogenic, as the name is, photo light chromo colorgenic mycobacteria form pigment only on exposure to the light. So these can be differentiated from the other mycobacteria on the basis of pigment production. Do or do not. There is no try. One should go for everything, and just do it. Other parameter is salt. A high salt or the sugar concentration will prevent growth and may kill bacteria. Halophiles or the salt-loving require high level of salt. Sodium chloride examples are marine prokaryotes survive well in the 3.5% salt. Some mesophile species are also salt tolerant, like Staphylococcus. This is very tough bacteria and it can tolerate the extremes of different physical and chemical factors. If environment have high salt, water will leave cell and cell would plasmolyze. There is principle of salting meat and other food products. So using sugar as preservative in the jams and jellies that preserve these products for longer time and can be used for halophiles. Here are the bacteria which are halophile. Osmotic effect. It is tolerance to the osmotic variation, pressure exerted by the weight of the water on cells and cell prevent rupture. Bacteria can tolerate osmotic variation due to the mechanical strength of the cell wall. If that is tough enough, that can able to tolerate the osmotic variation. Most of the bacteria are osmotically tolerant except for the Mycoplasma, Chlamydia and cell wall defective organism. Then osmotic effect on the bacteria is plasmolysis. When the bacteria are placed in a hypertonic solution, environment water leaves bacterial cell, cytoplasmic membrane shrinks and leaves cell wall, seen more in the gram-negative bacteria than gram-positive bacteria. Because from the hypertonic solution, the fluid will come out. Then plasmoptosis, sudden transfer of the bacteria from the concentrated solution, hypertonic to the distal water, which is hypotonic class plasmoptosis. There is sudden excessive transfer in of the water, leading to the swelling and rupture of the bacterial cell. Here one can see the plasmolysis and here hypertonic. The water, if these bacteria are exposed to the hypertonic environment, the water inside the bacteria will come out to the environment to make it isotonic. So if the bacteria are put into the isotonic media, the water from the outside that will go inside the bacteria and bacteria will swell up. And in this isotonic, some of the water will get inside and some of the water will come outside. So the condition will be isotonic. The next is hypotonic, which I was studying previously. That is, if the hypotonic environment is there, the water from the outside will get inside the bacteria. Here in isotonic, some of the water will go inside and some of the water from the bacteria will come outside to make the isotonic condition. But in hypotonic, the water will go inside, that will make swell the bacteria and the vacuole, and that is going to cause the rupture. This is plasmoptosis because of exposure to the hypotonic solution. Hydrostatic pressure, psychrophiles, as you know, these are the bacteria which can live in extreme cold. In deep ocean, waters are under extremely high hydrostatic pressure. In marine trenches, hydrostatic pressure is about 16,000 pounds per square inch psi. Bacteria that withstand this pressure called barophiles. These will die at the normal barometric pressure. The next parameter is mechanical and sonic stresses. Tough cell walls of the bacteria are ruptured by the mechanical stress like grinding or vigorous shaking with the glass beads. Exposure to the ultrasonic vibration also disintegrate the bacteria. So these are the barophiles which are able to withstand the extremes. And now, what is the way of the uptake of the nutrients by the bacterial cell. Bacteria use different transport mechanism. One is the passive distribution, very simple. Here molecules from the area of the high concentration, that is hypertonic, to the area of the low concentration, that is hypotonic. It requires a large concentration gradient for uptake, applicable to the limited small useful molecules like glycerol, hydrogen, water, oxygen, and carbon dioxide. Then facilitated diffusion, as the name is, this needs some facilitation to cause the diffusion. It involves a carrier molecule permeates to increase rate of the diffusion. There is a movement from area of the higher concentration to the area of the lower concentration, as in case of the passive diffusion, but there needs smaller concentration gradient than passive diffusion, and the rate plateaus when the carrier molecule becomes saturated. So here one can see lump of the sugar in a jar and sugar molecules that do travel here and do travel here and here. Ultimately, the higher concentration to the lower concentration, the molecules of the sugar will be equal. Here one can see the passive transport. One can see the transport molecules very easily, simply diffuse. But here there is one carrier molecule, permease, which pushes these molecules through this port. So that's why it is called facilitated diffusion. It needs the facility of the carrier molecule to come to this from outside to inside, and that is one directional from outside to inside, so it is called uniport. Then there is the active transport. Active transport needs energy requiring process of pumping or pushing the molecules and the oils across the membrane uphill against a concentration gradient, as opposed to the diffusion where is concentration gradient, but here is no concentration gradient, but you have to push the ions against the concentration gradient, that is from lower to the higher, where solute concentration is already higher than outside, but we have to push these molecules against this concentration. So that is active transport. There are three types of the active transport. Symport, transfer of the two substances in the same direction. Antiport, transport of the two substances in opposite direction, one coming in, other coming out. Group translocation, here molecules are modified as they are transported across the membrane just to get the easy passage. So here one can see the active transport, and here there is the concentration of the molecule is low here, but these are being pushed to the higher concentration through the energy ATP, and that is called active transport. Now symporter, as you can see, that is the two different compounds are coming inside the cell, and antiport, one is coming inside and the other is going outside. So here one can see the group translocation. There is binding, and that binding protein is just going to help the molecule to get inside, so that is the transport via the binding protein.